The overall goal of this procedure is to assemble a PDZ-Dependent Cystic Fibrosis Transmembrane Conductance Regulator, or CFTR, macromolecular signaling complex in vitro. To do this, the recombinant tagged fusion proteins are expressed in bacteria and purified. Next, cell lysates are obtained from cultures that express CFTR or CFTR binding proteins. Then, the CFTR and binding proteins are combined in vitro to assemble the CFTR-containing macromolecular signaling complex. Western blotting is then performed, which demonstrates the formation of CFTR macromolecular complexes and is then assessed via Western blotting.
The main advantage of this technique over existing methods is that we can directly detect and visualize the formation of a CFTR-containing triprotein macromolecular complex in vitro.
Though this method can provide insight into the formation of CFTR-containing macromolecular complex, it can also be applied in assembling non-CFTR-involved multi-protein complex, such as CXCR2 macromolecular signaling complex.
Most recently, using this same method, we have demonstrated that a chemokine receptor CXCR2 physically forms a protein complex with the downstream effect PLC beta 3, mediated via the nerve Wang in neutrophils.
Using PCR, amplify the DNA that encodes the last 50 to 100 amino acids containing the PDZ motifs at C-terminus for the cAMP transporter MRP4 and the PDZ1 domain of sodium hydrogen exchanger regulatory factor, or NHERF. Next, clone the MRP4 PCR products into PGEX.